Stanford University.
The privilege of running a program that really, I think, brings together a lot of what you've been talking about this week with things this afternoon as well, which is to try to use the really enormous advances in understanding of the human genome and human genetics in the last decade or so with ideas about screening, early detection, and some of the things we're talking about today, to think about risk assessment and how one can use those sorts of tools in very high-risk people of genetic predisposition to cancer to try to decrease incidence and deaths of cancer. And so this is yet another version of what you've seen from several folks today. So this is SEER data from SEER data looking at deaths from cancer across much of the last century, and lets you say a number of things. Some obvious things that we know, that lung cancer rates, this is in women, that lung cancer rates are increased in conjunction with smoking rates, that stomach cancer deaths came down significantly with changes in food and refrigeration. But another couple interesting things. So, for example, colorectal cancer rates have been slowly decreasing for quite a long time. And then this, what we've been talking about a lot, this change in the slope of the breast cancer. Now, this is death rates, not incidence, starting in the mid 85 to 90 range. So this sort of change lets you think about what are the factors that are going into that. I mean, we talked a lot about estrogen use, but that effect on incidence isn't really probably had little effect here at these times. And if you start to peel apart this a little bit more carefully, this probably has something to do about half with better treatment, though treatment options in breast cancer have been incremental and modest, but it has impacted survival, and half screening. Because about the time this started to change was when mammography became much more important. And so this leads to this paradigm of though the cancer war for the past 3 decades has been largely focused at the research level on identifying better treatments, ways to deal with metastatic cancer, and has been, modest in its success. In many ways, one can think that focusing at earlier stages in the progression of normal tissue to metastatic cancer might have a larger impact, that is, on early detection or ways to prevent cancer in the first place. But of course that's very difficult to do and is a much larger population of people than those that are presenting with cancer, and so there's lots of issues in terms of cost and access to these. And so a major area that we're very interested in then is risk assessment, in our particular case genetic risk assessment, to try to identify those individuals who are at the highest risk, albeit a small percentage of this total population, for whom then one can focus some of these newer technologies, particularly in early detection and hopefully ultimately prevention, more successfully. So for us, that's then all about DNA, and of course, as we've talked about all week, cancer is a genetic disease. Cancers all have multiple mutations in multiple genes, and so one of the basic underlying defects, of course, is genomic instability and how cells lose that maintenance and result in that. And of course, you've talked lots about the types of genes that are involved in genomic instability this week. But an interesting area when one starts to talk about hereditary cancer syndromes is that this is an ideal place to start to study some of those genes and what drives that early genomic instability. If one can identify those genes conferring a familial susceptibility to cancer, they are often those very genes that are involved in early genomic stability and lead to the multiple mutations that actually are required for an invasive cancer. So for today, I want to talk about hereditary cancer syndromes, which we define as primarily autosomal dominant familial patterns of inheritance. And we'll talk about that more specifically, due to germline mutations that we can oftentimes identify, and that probably accounts for about 5 to 10% of all solid common tumors. And I'm going to talk specifically today about breast cancer and colon cancer, though we could talk about many other cancers in the same situation. Talk about how advances in clinical genetics allow for genetic testing to identify these individuals at high cancer susceptibility, and a little bit about how genetic testing takes place and the sort of clinical context we use, and then finish by giving some examples about how a really multidisciplinary approach to early detection can make significant inroads now into decreasing death using screening, medical, and surgical preventative techniques. So about 10 years ago now, we actually started a dedicated clinic at Stanford, one of the earlier clinics to do this, focused on the identification and genetic testing and counseling of families at high hereditary cancer risk. And so over the years, many more syndromes have been identified, and this is just a partial listing of the sort of patients and families we see with risks to these many different kinds of cancers and the genes that have now been identified that, when altered in the germline, confer that risk. And so for today, I'm going to, as examples, focus purely on hereditary breast cancer due to BRCA1 and 2 that we've talked about already, and hereditary colon cancer, often called HNPCC, or hereditary non-polyposis colon cancer. But the principles in these two really apply to all the cancer families that we see. So we use these definitions to start to think about who falls into some of these groups, which is that as opposed to sporadic cancers, which probably count for 2/3 or so of cancers that are occurring at the common rate in populations, the different populations that are looked at, familial cancers are those that seem to have some sort of clustering in families at a higher rate than one would expect from the population-based incidence. And that probably accounts for 20 to 25%, maybe 1/3 of cancers. But hereditary cancers we're defining as those 5 to 10% in which we really have identified a germline mutation, have a recognizable pattern of inheritance, usually autosomal dominant, and often have a very early age of onset, have multiple primaries in individuals, and for whom now we can most often identify germline genetic alterations that explain those families. So this is sort of the basis then really of the difference between hereditary and non-hereditary cancers. It's often called the two-hit model, or the Knudsen hypothesis, which is that for many of the genes involved in cancer development, tumor suppressor genes, DNA repair genes, genes involved in genomic instability, a cell needs to lose both copies or inactivate function of both copies of those to lead to genomic instability or lead to a cancer phenotype. Meaning at the cell level they're recessive. One copy is often adequate to maintain a normal function, like a cell cycle regulated function. And so sporadic cancers then most often need to lose both copies of that particular gene at that particular locus in a cell to lose function of that gene. And then of course probably multiple genes need to be inactivated in similar manners to truly transform a cell. And so statistically that's a pretty unlikely thing to happen, why not more cancers are actually occurring. In the hereditary situation, the underlying paradigm is that a gene has been mutated in the germline, meaning it's gotten an altered version has gotten passed along through egg or sperm cells so that a progeny has inherited that mutated gene in every cell of their body. So of course they're far more likely then to lose the second copy or have a second hit in some cell in their lifetime than in this situation where that has to happen twice. And so this explains why these cancers are happening early, why they happen so often, why often in these syndromes nearly every individual who inherits that change will get a cancer, and why they can often happen in multiple tissues or multiple times in a person's life because all those tissues are at risk. What it doesn't really explain is why these different syndromes have these different patterns of cancer, why BRCA1 and 2 mutations conferring risk for breast and ovarian cancer, but not for other cancers. And similarly, since all these tissues seem to be at risk, and we really don't have a very good explanation for that. Sorry. So this also explains then the patterns of inheritance you see, which is like any dominant genetic pattern of inheritance, there's a 50-50 risk of passing on the altered allele with each new child. So in this situation, the red allele in the father then is abnormal, and the mom has two normal copies of that. So with each child, there's a 50-50 risk, independent of each other, that abnormal copy will get passed on. And so this is classic autosomal dominance, and in many medical genetic syndromes, that means that 50% of those offspring will get whatever the disease is. In cancer susceptibility, remember, the trait that's getting passed on is susceptibility to cancer, so cancer risk. And we'll come into this in a minute, because since it's not 100% penetrant, it doesn't necessarily mean every one of these people will get it. It just means they're at a much higher risk, and so the family pedigrees can look kind of complicated. So in a purely autosomal dominant syndrome, you tend to see these characteristics, that you see vertical transmission of the trait, the genetic trait, in multiple generations, that you see about half of each generation will express that trait, and that it's not a sex-linked condition. It's seen equally in men and women. But in these cancer syndromes, it more often looks like this, where now orange is showing affected with cancer, and you're seeing it in multiple generations, and you're seeing it maybe in about half. But here it looks like there's a skip generation. The grandfather here got cancer, the grandkids, multiple cases of cancer, but nothing happened here. And similarly here, there seems to be a skip generation. And so that can be due to lots of reasons. Some of them are this. One is that the penetrance of getting cancer is not 100%, and there's age-specific changes in that penetrance. So this could just have been an obligate carrier who passed on that genetic susceptibility to cancer, but she herself has not developed it or may develop it in the future. The other thing is cancer is common. And so this just shows a typical plot of age-specific penetrance, meaning that even in individuals who've inherited a gene causing a very high cancer susceptibility, the chance of getting cancer often doesn't increase till 20s, 30s, 40s. It may be a very high lifetime, in this case an 80% lifetime risk of cancer by the age of 80, but it changes over the lifetime. And so again, when you're looking at family trees and talking to families, you have to take into account the age of those individuals. Well, what affects penetrance? Many things. Certainly that susceptibility is going to be modified by the environment and by interactions that individual has, and so clear-cut things like carcinogens from tobacco smoke, hormonal factors that alter carcinogenic rates, as we talked about today, but also modifier genes. So the the thousands of other genes that individual inherits is going to modify the expression of that genetic risk. And so this is why we really don't have a good grasp yet on why, if you look at families, you can see in the same family with the same mutation in individuals, some will get cancer, some won't. The age at which they get it will be different. The type of cancer they get may be different. And so that may be driven to some extent by chance, but it probably is driven by a large extent by co-modifier genes that are getting inherited in a very complex way, not in a Mendelian fashion as these genes are. And so at this point for us, it is difficult to actually diagnostically predict, but that's a major area of research interest. Well, so given that background, then when one thinks about clinical genetic susceptibility syndromes to cancer, here are really the three questions that I think you have to hold each up to. Who should you test for genetic susceptibility, meaning how do you find these families? What are the criteria one uses? What are the actual risks of cancer associated with known mutations that we can identify and communicate to people? And then, of course, most importantly, what can actually be done to prevent cancer in unaffected carriers who one can identify? This is obviously the clinical importance to this whole exercise. Well, when one is clinically doing this, there's a few rules that our genetic counselors always try to start with, which is when one's looking at a pedigree like this, you always want to try to start in terms of thinking about genetic testing with an affected individual, meaning affected with cancer. A very, very common scenario is we see a family like this, colon cancer, and three individuals, all pretty young, 42, 45, 45. So that makes one really think about a susceptibility syndrome. But actually, the granddaughter here comes to genetic counseling and asking, what's my risk of getting colon cancer based on my family history? Well, her risk is certainly substantial, But if you have not identified the genetic change that's driving this, it's hard to counsel her, because if we find it in her father, then we can test her, but of course she had a 50-50 chance that she would get it or not get it. So if we started testing her and did not find a problem, that doesn't let us rule out that there's a genetic susceptibility going on in the family. It also doesn't let us rule out that it's a gene that we can't find. So it's a non-informative test. So whenever possible, we try to start with an affected. But of course, because this is cancer, many of those affected individuals will have died and will not be available. And so we often are left in this situation. And so it's one aspect of genetic counseling that's important to explain to people the limitations of what that test can inform them. Another important principle is how to interpret negative results, and this is getting very confused now as genetic testing is sort of filtering out and getting done in primary care clinics and such, which is that a negative test does not necessarily rule out risk. So again, here's a situation, same kind of family with multiple cases. A true negative would be this situation where we began testing with an affected and actually found a mutation. This is a gene we'll talk about a little bit later, MSH2. So this 45-year-old woman was found to have an MSH2 deleterious mutation. Then our patient got tested and he did not have it. So that's a true negative. We know he was 50% did not inherit it. But again, just like the last slide, if we start here and simply don't find it in that individual and had not tracked it down in the family, that's an inconclusive test. It's not negative. And a lot of folks just get tested now, get a negative test, and presume their risk is low. In fact, this person should be clinically told that they're at an elevated risk for cancer and proceed with that screening, even in the absence of a genetic of an underlying genetic susceptibility. So let me first talk about genetic susceptibility to breast cancer, since we talked about it a lot this afternoon in other examples, and then I'll go ahead and talk about colon cancer a little bit. Well, as you've heard, there's lots of risk factors for breast cancer. Aging, family history, which is what I'll focus on, a variety of hormonal changes, estrogen use, we talked about, dietary factors, so complex analysis as the discussion has revealed in thinking about these things. But family history is one thing that definitely drives breast cancer risk. And so this is the kind of remarkable families we see in this clinic. So here's a 3-generation pedigree of a family that you can see was breast cancer in 3 generations. A woman who died at the age of 30, or diagnosed at the age of 31 with breast cancer and died. She had twins, identical twins, who were diagnosed with breast cancer at 26 and 29. One of their daughters came to see us, who had unfortunately been diagnosed with breast cancer at 30. And so this is a classic BRCA1 mutant type family. In fact, that's what was found, a BRCA1 mutation, which clearly was explaining what was going on here. The question really is then, how does this affect what we think about her for her future treatment, and what could we do to potentially test and do better for her sister in terms of protecting her from developing breast cancer? Well, if you look at rates of incidence of breast and ovarian cancer in the U.S., you can see, again, about 3/4 or so seem to be sporadic, and about 1/4 or so seem to have some familial or hereditary basis. We think about 5 to 10% are driven by BRCA1 mutations. And so since breast cancer is very common, this makes up a substantial number of deaths in the U.S., 40,000 or so a year. Ovarian cancer, similarly, about 10 or 15% seems to be driven by hereditary BRCA1 mutations. Ovarian cancer is less common than breast cancer, so the overall numbers are smaller, but the rates are quite similar. The majority of genes that we know that cause hereditary breast ovarian cancer syndrome are BRCA1 and 2, but that's not all. And there's certainly rare cases of p53, PTEN, and others causing similar syndromes. But if one adds that up, there's still a pretty big chunk that's unexplained. And so there's probably a number of genes that remain uncovered that contribute to breast cancer risk, though maybe not at the penetrance that we've seen. If you look at the incidence, so in Caucasian populations, about between 1 in 100 and 1 in 400 women overall will be carrying BRCA1 mutations. Pretty substantial. And if you look in Ashkenazi Jewish populations that carry founder mutations that were selected a millennium ago, and they exist at a higher incidence. Up to 1 in 40 women of Ashkenazi descent will be carrying one of the BRCA1 or 2 founder mutations. So a substantial number of people. Estimates are in this country that up to 500,000 women are actually carriers for whom these sorts of advances are relevant. So how do we find these women and these families? Well, clues like this: multiple cases of early onset breast cancer, ovarian cancer in a family that has any breast cancer at all is a very high risk, or particularly in the same woman. Bilateral breast cancer is high risk. Simply being of Ashkenazi Jewish heritage means your risk of carrying BRCA1 or 2 mutations is tenfold others, as I just showed you. Male breast cancer, which is quite unusual, but is much more common in these families with genetic susceptibility, is another thing we factor in into identifying high-risk families. So what is the risk then? Well, for a woman who's been identified with a BRCA1 germline deleterious mutation, her risk of breast cancer is up to 60% by age 70. Her risk of a second primary breast cancer is nearly the same, 40 to 60%. And ovarian cancer is quite high as well, 30 to 40%. BRCA2 has slightly different risks. The breast cancer risk is currently estimated up to about 50%. The ovarian cancer risk lower, in the order of 10 to 20%. But male breast cancer is far more common. About 6% of these families will have male breast cancer, which is in far excess of what one sees in the population. And then there's a number of other cancers that seem to have a small increased risk, on the order of 1.5 to 3-fold, particularly pancreatic cancer and melanoma, which we actually are starting to think about in terms of screening. So here are the actual then age-specific penetrance risks by BRCA1 or BRCA2 for breast and ovarian cancer. And so this just adds up to the lifetime risks that I just told you. But what's important here is seeing when these risks start to increase. So you can see for BRCA1, the breast cancer risk really starts to go up at about the age of 30, whereas the ovarian cancer risk doesn't start to increase really till about 40. Whereas for BRCA2, the ovarian cancer risk is almost 50 when it starts to go up, and of course it's not as high overall. And so it's these sorts of curves that we use to try to provide counseling in terms of what types of interventional maneuvers we'll do, and we'll talk about those in a minute. So that mutation penetrance, then what explains that? And as I said, we don't have a great idea, but we certainly know that there's great variations not only between families, but within the same family that are carrying the exact same mutation. And that the penetrance for breast versus ovarian cancer is quite different. So is that an influence of other genes that are shared in the family and there might be different between individuals? Is this differences or similarities in exposure and environment? And no doubt it's some of both, and it's hard to pull those apart. There's data that there's variation in penetrance by birth cohort within families with the same mutation. So breast cancer risk actually increases up to 4-fold in younger individuals or cohorts of younger individuals in families than older in the same family. And why is that? Potentially shared lifestyles and changes in reproductive exposures, hormonal exposures in different generations. We don't really know. And then, of course, how much do genes and environment modify BRCA1 and 2 risk? Clearly, all the risk factors for breast cancer are modifiers for this already elevated risk due to BRCA1 and 2. But how much of it is, and if one can modify those, is unclear. And so there's large cooperative groups now trying to get at those questions. There's certainly ethnic influence on BRCA1 and 2 mutation risk for breast cancer. We talked about the number of carriers in the United States, between 250 and 500,000, and the carrier rates in different populations. But the rate of carrying BRCA1 and 2 mutations is different in different ethnic populations, not surprising. In fact, Hispanics seem to have a higher rate of BRCA1 mutations than African Americans than Asian Americans, whereas for BRCA2, that difference is actually flipped. We just published a paper this week with Dee and others here looking at BRCA1 and 2 mutations in Asian Americans on the West Coast and Hawaii, and actually found that the models that we use to predict BRCA carriage dramatically underpredict, particularly BRCA2 mutations. Actually, Asian women are carrying twice as many BRCA2 mutations based on their breast cancer risk than one would predict. So all of genetics is very heavily based on Caucasian populations at this point, and so the risks and the penetrance of genetic mutations in other ethnic populations are only just beginning to become explored. That's going to make a major shift in risk prediction. And I think this is just saying what I just said, that the incidence of these mutations, as well as the penetrance of getting cancer should one inherit that mutations, are different in different populations, and we still don't know enough about it. Well, how does one actually then go about counseling women about these mutations and actually thinking about management in them? And so this counseling aspect is really key to the genetic testing process, which means that before you even get into this, we meet with these families and these individuals to talk through basically what we just talked through here. What are the principles of genetic testing? What are the pros and cons of doing it, and what are the limitations of the knowledge that you might get out of it, so that people really have an informed consent, essentially, are informed as to those issues before they even discuss having a genetic test. And then we always meet with individuals, even if they're negative for those tests, after genetic tests, to again really go through what one can say with certainty about those tests, and certainly if they're positive, to then begin discussions about interventions and management goals. So in terms of BRCA mutations, when one tests for that, one does full gene sequencing. These are big genes, so it costs a lot. It's about $3,000 to sequence BRCA1 and 2 through commercial clinical labs. The detection rate is high. It's actually better than this now. We also do large rearrangements, and that brings us up to about 95%. So the testing is quite good, but there's certainly issues that we can talk about later of reimbursement and access to these tests. But clinically, what can one do? So if one identifies a woman at high risk for breast and ovarian cancer due to a BRCA1 or 2 mutation, there's a number of proven interventions now that are important. Clearly, a prophylactic mastectomy dramatically decreases the risk of breast cancer in a woman who's carrying a BRCA1 or 2 mutation, a very unacceptable approach to most women and most of us, and yet until recently, really one of the few that had a major impact. We'll talk about surveillance with imaging and tamoxifen in a minute. Prophylactic oophorectomy not only significantly reduces the risk of ovarian cancer, but also reduces the risk of breast cancer by about half when done in a premenopausal woman. So the general recommendations for us then are prophylactic oophorectomies actually at age 40 or after childbearing years, since it has a clear impact on both breast and ovarian cancer risk, and we have very poor tools for screening for ovarian cancer, but then a personalized approach to breast screening, either through surveillance and or consideration of mastectomy. Well, the problem with surveillance in this population is that mammography is not a great tool for picking up breast cancer in young women, particularly who have BRCA1 and 2 mutations, for a variety of reasons. We talked about breast density and detection. BRCA2 associated tumors have certain characteristics that make them particularly difficult. There's issues about radiation exposure in this population. These genes are important in DNA repair of radiation damage, and so long-term exposure to mammograms may be of concern. So a major advance then in this population has really been that of breast MRI, or magnetic resonance imaging. So this is an example then comparing imaging from the same woman who had a mammogram. This was a young woman with a BRCA1 mutation, and this is a normal mammogram. So we don't see any spots or white lumps that we would think of as cancer here, whereas the MRI on the same woman done at the same time shows this clear abnormality, even without this huge arrow pointing at it. We could see that. And you could see how that's easily missed on a mammogram of this woman with fairly dense breasts, so you see a lot of these white areas. So MRI has been shown to pick up breast cancers earlier and with more sensitivity in this population, and is now the standard of care really in BRCA1 or 2 carriers, and even women at elevated risk simply based on family history. And I'll come back to how that's made an impact on decisions about prophylactic mastectomy. Is that screening good enough that one can feel comfortable with that? Well, what about chemoprevention? You heard about from Marcia about trials of tamoxifen in reducing breast cancer risk, which it certainly does, but we really don't know if tamoxifen is decreasing risk in BRCA1 and 2 carriers, and that's because in those trials there's a very small number of these carriers, as well as theoretically most of the cancers that are occurring in this population are actually ER negative, and one might not expect that tamoxifen would have such an effect there. In point of fact, tamoxifen has been taken up by very few women for breast cancer prevention in general, and so very few of these women are really pursuing that, probably appropriately so, since we really don't have data on it. This is, just to finish this part, this is a study, actually Sylvia Plavridis, who you heard from this morning, and Alison Curran in my group did, to try to model the effect of various risk-reducing options on lifespan in women. So this is an area that there's never going to be a randomized trial. No one's ever now going to do a randomized trial of breast MRI versus nothing in a woman at very high genetic risk, because it would be unethical, of course, to randomize somebody to the arm that didn't have the intervention. And so it will be, and the same thing with all these other interventions. So this is one way to try to get at looking at this. And so this is looking at the average age of lifespan of a BRCA1 or 2 carrier who had no screening, and you can see that, and actually in the paper there's about a 40% decrease in average lifespan in that woman, versus those that pursued mammography alone. This is starting age 25, mammography plus MRI, prophylactic mastectomy at age 25, prophylactic oophorectomy at age 40, or all of these things. And of course there's an incremental increase in all of these. But what's particularly important is that though prophylactic mastectomy certainly has an effect, doing screening in the absence of a mastectomy with an oophorectomy at age 40 seems to do similarly or even better at improving lifespan. So this is certainly going to help in terms of thinking about options that women want to take based on their risk assessment and their personal choices. Okay, so I want to finish for the last few minutes talking about hereditary colon cancer. Lots of the same principles, but a very different type of syndrome. So here's a typical pedigree of a family we'd see. Gastric cancer, endometrial cancer, colon polyps, and this gentleman, 32-year-old, came to see us. We wouldn't necessarily think of a colon cancer syndrome, but it turns out actually that's what this is. So just to talk about colon cancer for a minute, very common in the West. In populations, the lifetime risk is on the order of about 6%, and most of these are occurring in individuals over 50. And important to colon cancer, that's different from other cancers, is that it arises from polyps, which we'll come back to as something we can sample. And so here again, it's just showing the effect of family history on colon cancer risk, and with additional first and second degree relatives, of course, that incidence goes up. We're going to talk about this syndrome here, hereditary non-polyposis colon cancer, with a very, very high lifetime risk of colon cancer. Well, this is the so-called Vogelgram, or mapping out the multi-step changes that occur from normal colon epithelium through polyps, really a preneoplastic tissue, to invasive colon cancer that was allowed by being able to sample polyps through colonoscopy. So that's a really unique situation compared to a lot of other cancers where you don't really have access to those preneoplastic tissues. And the point here really is that it requires multiple mutations to get to an invasive cancer, and there's probably some pattern of these, but the earliest ones tend to be those invoking genomic instability. APC we won't talk about today, but in the germline causes very rare familial adenomatous polyposis, actually shown here. So if you look at all colon cancer, again, about 2/3 is sporadic, and a third or so has a familial basis. And then there's these various genetic syndromes, rare FAP and other syndromes, and I want to talk about this 5% or so driven by mutations in mismatched DNA repair genes. So if you come back to this multi-step carcinogenesis idea, about 80% of colon cancer seems to be due to this APC KRAS-driven pathway, but a substantial proportion, 20% or so, is due to a microsatellite instability pathway, when due to early changes in genes that are involved in DNA mismatch repair, MSH2, MLH1, and others. And this results in a different kind of chromosomal instability. You see less aneuploidy and chromosomal breaks, and more what I'll show you actually is referred to as microsatellite instability. So this was, we learned about this from a syndrome called Lynch syndrome, named for Henry Lynch, one of the real early advocates of cancer susceptibility syndromes and identifying them. And so this is an early colon cancer syndrome. The tumors tend to be in the right side of the colon, which is not true in sporadic colon so much. But importantly, if one thinks about that pedigree I just showed you, that there's other cancers in these families. In addition to colon cancer, there's a lot of endometrial cancer, ovarian cancer, GU tract, GI tract, and others. So we have to look at this sort of whole set of cancers, but they occur in that same autosomal pattern of inheritance that we talked about. And criteria were set up for these. This actually predated no understanding the genetics, so this is the state of medical genetics really prior to more knowledge about the human genome, which is that high-risk families, such as 3 or more cases, and one is particularly young and a first-degree relative to the other 2. If you look at these about 2/3 of these families, you could identify a germline mutation in one of a set of DNA mismatch repair genes. And so the genetics is that, again, it's an autosomal dominant syndrome. The penetrance is about 80% for colon cancer. I'll show you what it is for the others in a minute. And this set of 5 genes predominantly drives these, though mutations of these 2 are the most common. And so that's just demonstrated here. So again, if one looks at this slice of colon cancer that's HMPCC, about 1/3 is due to MSH2 mutations, 1/3 MLH1, some other rare ones, and then about 1/3 that we just simply don't know yet. So presumably other genes that have not been identified that are leading to a similar syndrome. And I want to just quickly think of the mechanism of mismatch repair, because it actually explains the diagnostic test that we can use to actually phenotype these cancers. So this is just showing these sets of mismatch repair proteins which function as heterodimers to actually monitor double-stranded DNA for mismatches. So this is simply replication errors that occur at some low but real rate every time a cell divides, and if not corrected will of course lead to a mutation. And so these proteins serve to identify that mismatch, to actually excise a DNA stretch containing it, and to lead to repair replication and resynthesis of a normal strand. So if any of these proteins go awry, or the genes are mutated, these types of mismatches will accumulate. And they particularly accumulate in microsatellite sequences. So that's just defined as a highly repetitive repeat of a certain stretch. So for instance, a dinucleotide repeat of 20 or so here has a high likelihood of slippage during replication and adding in extra base pairs, because everything matches up and downstream of that. The mismatch repair system recognizes that, repairs it, but if it's defective, it's not going to repair that, and with rounds of replication, you're going to see extension or contraction of these links of microsatellites. So the clinical test for, quote, microsatellite instability is simply to PCR amplify selected microsatellite regions and look for banding around those loci, as opposed to stable microsatellites in tumor tissue versus normal tissue. So this is actually what we do. And so this is an example of doing microsatellite instability testing on tumor versus normal tissue, like a blood sample, in a number of loci. And if 2 of the 5 show MSI, that's defined as MSI high, and that's very predictive then of hereditary non-polyposis colon cancer, and would lead us to go on and actually think about genetic testing. Well, so what is then the risk of cancer? Well, again, about an 80% lifetime risk of colon cancer, but a very substantial risk of a lot of other cancers, particularly endometrial cancer in women. And can we do anything about it? Well, here again, there's definitely interventions that have been shown to decrease mortality, actually. So colonoscopy, regular colonoscopy surveillance has statistically improved survival in individuals with HMPCC compared to those that do not undergo surveillance. And a hysterectomy, of course, to remove risk of endometrial cancer, has significantly decreased the incidence of developing endometrial cancer, though this is not showing an effect on survival at this point, and those studies aren't mature enough actually to show that, but it is a choice some women make at an appropriate time. So if you come back to our family then, So we thought about HNPCC because of this cluster of gastric cancer, endometrial cancer, and colon polyps in somebody who's pretty young, even though there wasn't a colon cancer in this family. And indeed, that was the case. And so by testing an affected, so we tested our patient's mom who had endometrial cancer. She in fact showed microsatellite instability if we looked at her tumor block, and then by germline testing was found to have an MSH2 mutation. And of course each of the kids then had a 50-50 chance of inheriting that. Our patient did inherit it. His SIBs were similarly tested. One did and one didn't inherit it. And so now none of them have cancer. But the two who have high susceptibility, of course, will have regular colonoscopies. The women will have regular gynecologic exams, and hopefully any cancers that develop will be caught early enough that it's at a very curative stage, and they won't end up with invasive cancers like the older generations in the family. So I think I need to finish up. So just to come back to the point that there's lots of other syndromes, lots of other germline testing that can be done. So the principles are very much the same. And so in terms of the clinical aspects of who should receive testing, it really is somebody who has a reasonable likelihood of carrying an altered cancer susceptibility gene based on those predictors that we talked about, that a genetic test is available, that it will actually influence medical management at some point, and that the patient wants the information and will do something positive, hopefully, for them with it. These are all the other syndromes. And just to make the final point in this era, that genetic susceptibility testing for cancer or other things is not a population-based screen. This has to be done in the context of thinking about risk and comprehensive plans for managing that risk. And so I think it's very important that genetic counseling is a component of genetic testing. And so I'll finish with this idea that we worry now that it's all in the genes, it's all just a barcode, and it's completely impersonal. No, just lie back and we'll scan the barcode. And so that's true. DNA is a barcode, but it's incredibly important that those changes, that variation, is thought of in context of medical management and risk, so that we can benefit patients while learning from genetic sequence to help us. And so this is our group. If those are interested, you can always refer families to us at a central number. Elton Crane and myself are the oncologists that run the GI and the breast portion of that group. Nikki Chun and Carrie Kingham are genetic counselors who specifically see these cancer families and really do a terrific job in explaining these kinds of complicated risk decisions to them and ideas about genetic testing. And with that, I'll finish and take any of your questions. Thanks.
For more, please visit us at stanford.edu.